Severe congenital heart defects are a life-threatening condition that requires complex care and invasive surgery. Over the last decades, advances in cardiac surgery, neonatal care, and improvements in understanding the condition have allowed most of these infants to survive. Unfortunately, some of them suffer comorbidities, such as brain injuries that can lead to neurodevelopmental impairments that can be preventable. The TinyBrains project focuses on understanding the cellular origin of a certain type of brain injury that may happen because of hypoxic ischemic events that occur during surgery. The goal is to understand if they have an effect beyond the local brain lesions and if they also affect the neurovascular unit. As neurons do not have internal reserves of glucose and oxygen, their activation requires the quick arrival of more nutrients, including oxygen. The neurovascular unit acts as the link between neuronal function and oxygen metabolism, as its components collectively regulate cerebral blood flow to deliver the requisite nutrients to activate the neurons. In a process known as the hemodynamic response, the neurovascular unit ensures that blood delivers enough oxygen to active neurons that require more energy than the inactive ones. Although the neurovascular unit is crucial for seamless brain activity since it supplies the required energy, there are no practical and comprehensive tools to evaluate its function in neonates, as current technologies are not suitable for cot-side use, making them difficult to utilize in congenital heart defect patients. To fill this gap, TinyBrains provides a neuroimaging device to monitor the neurovascular unit before and after invasive treatments, with the goal of using this information to help doctors improve neuroprotective strategies in this population, preventing the development of learning disabilities, poor school performance, and other neurodevelopmental constraints that congenital heart defect patients, their families, and other caretakers face. TinyBrains combines advanced biophotonics technologies and electroencephalography in a research tool to simultaneously measure cerebral hemodynamics, oxygen metabolism, and electrophysiology. To provide this information, TinyBrains has integrated on a single device electroencephalography, diffuse correlation tomography, and near-infrared diffuse optical tomography. Electroencephalography, or EEG, measures the brain's electrical activity using electrodes attached to the scalp. As neurons communicate between them through electrical impulses, the electrical activity is linked to the activation of specific areas of the brain. EEG is widely used in cognitive neurosciences to understand normal brain functioning, research learning mechanisms, and detect neuronal alterations, such as epilepsy. Functional near-infrared spectroscopy is a non-invasive optical technique that uses near-infrared light, which offers deep penetration within the tissue to measure oxygenated and deoxygenated hemoglobin concentrations within the brain. This information is obtained non-invasively, thanks to the relationship between oxygen consumption in the brain and brain activity. Blood with a higher concentration of oxyhemoglobin enters the activated region, and as neurons consume the oxygen, the oxyhemoglobin decreases and the deoxyhemoglobin increases. As oxy and deoxyhemoglobin have different colors, the light is absorbed differently by the blood before and after oxygen consumption, so the oxygen consumption in the studied areas can be tracked by using their absorption spectra. Diffuse correlation spectroscopy, or DCS, is a non-invasive optical technique that takes advantage of the coherence of laser light to quantify the blood flow in the brain. It is used in cognitive studies since it provides real-time information about the response of the brain hemodynamics. Diffuse correlation spectroscopy detects how fast the scatterers, the red blood cells, move by looking at the statistics of laser speckles. When the light flickers rapidly, it indicates that the scatterers are moving quickly. It is done by characterizing the laser speckle intensity distributions and using a physical model to link it to the blood flow. Both fNIRS and DCS can be measured tomographically by a spread of sources and detectors, and the information retrieved for each can be combined to form three-dimensional representations of cerebral hemodynamics. The tomographic approach is only possible if the sources and detectors are arranged very closely to each other. When the probe is set out in this way, it is called high density. In summary, E gives information about neuronal activity, fNIRS provides information about oxygen delivery and consumption, and DCS characterizes the blood flow in the brain. TinyBrains combines these techniques in a high-density probe, a head cap with an array of sources and detectors that cover a significant area of the neonatal head. This arrangement allows obtaining topographic 2-dimensional and tomographic 3-dimensional reconstructions, visually representing the parameters of interest. The device provides a cortical mapping of the neonatal brain that has not been available for clinicians until now. The tomographic reconstruction will have specificity because it can distinguish the depth of the activity and localize it, allowing for a better characterization of its amplitude and temporal evolution. TinyBrains scans, in 2 seconds, the areas of interest in the temporal and the parietal lobes. To investigate how the vascular network adapts to neuronal activation, infants are exposed to an auditory stimulus before and after the surgery, measuring evoked potentials. By doing the recording twice, researchers see if a low blood flow alters the cellular mechanisms neurons use to process information, which will be shown with changes in the evoked potentials, providing information about the neurovascular response in neonates. Preliminary results show that optical technologies can be successfully used in congenital heart defect patients, providing crucial information to improve management strategies or create neuroprotective agents. The lack of neuromonitoring tools for neonatal patients makes it difficult for practitioners to find adequate neuroprotective strategies in congenital heart defect patients. The team has successfully merged EEG, fNIRS, and DCS in a high-density probe fit for neonatal head sizes. TinyBrains provides non-invasive imaging of the brain that can be used to study neurovascular coupling in immature vascular systems, as well as the impact of invasive treatments in the first weeks of life.